Namaste friends, welcome to NEP Solutions. So friends, in this video I am going to explain the very important geology concept, that is TRP operon or repressible operon. So here it goes. First we will learn about the definition. The TRP operon is a gene segment that is responsible for the regulation of the synthesis of amino acid tryptophan. Trypoperon is a gene segment. It is a gene segment. It is responsible for regulation of the synthesis. What it synthesizes? It will synthesize the amino acid, that is tryptophan. Because of this, it is called trypoperon. TRP. TRP means tryptophan, that is the amino acid. Then the TRP operon consists major gene. We all know that in lac operon it has only 3 genes, major genes, structural genes, that are ZYA. But here the matter is very different. It consists 5 major genes such as TRYP-E, TRYP-D, TRYP-C, TRYP-B and TRYP-A. But here they code for only one enzyme, that is tryptophan synthetase. Trypoperon is activated upon low concentration of the amino acid tryptophan in the cell. It will activate when there is a low concentration in the tryptophan in the cell. Charles Yanofsky and his co-workers studied the role of regulatory and structural genes of the trypoperon. These are the two scientists. He is a scientist. With his co-workers or colleagues, he studied the trypoperon. Trypoperon, it is normally on mode. It has 2 modes, that is on mode and off mode. As usual, we have know that. So the first mode or basic we will see here. This is the diagram. The sequence consists of 2 parts, that are regulatory gene part and structural gene part. This is the regulatory gene part and that is TRYP-A to E. These are the structural genes. Let us study one by one. Regulatory gene part contains P refers to promoter and R refers to regulatory gene and O refers to operator. Then the tryptophan operon consists of 5 major structures, as I already told that, structural genes. And this is the diagram. In on mode we are learning here on mode. Here we have to write RNA polymerase. RNA polymerase will attack here and here also. RNA polymerase enzyme attaches promoter region. When RNA polymerase slides over the repressor gene, the repressor gene undergoes transcription and translation to form a protein. When it attacks the promoter, repressor regulatory gene produces protein. By the process of first transcription happens, then translation. You all know that. Then the formed protein act as apoenzyme. On its own, apoenzyme will not work or function. Hence it is inactive form. It is inactive because it acts as apoenzyme. When RNA polymerase attaches another promoter region, it slides over. When another RNA polymerase attaches to the promoter, it slides over. Now operator region is free. The operator gene allows process in order to synthesize enzymes. All the 5 structural genes of tryptophan. All the 5 structural genes synthesize a common enzyme, that is tryptophan synthetase. Tryptophan synthetase enzymes helps in formation of tryptophan amino acid using raw materials. Now the required amount of tryptophan is synthesized and it is utilized by body for growth. The on mode of tryptophan operon turned off when an extra tryptophan is synthesized. It will work until the body gets required amino acid. When any one amino acid is synthesized more, it will turned off automatically. Then now we will see how it will get to turned off mode, means off mode. When RNA polymerase enzyme attaches to the promoter region, then RNA polymerase slides over the repressor gene and undergo transcription and translation to form a protein. This protein is normally inactive form. As soon as the extra tryptophan attaches the protein, it forms a complex and the complex become active. It will become active when an extra tryptophan attaches to it. In on mode, the synthesized protein acting as apoenzyme, and in off mode, as soon as the protein binds with an extra tryptophan, it acts as holoenzyme. Remember the difference. Thus the complex get activated. When it started to act as holoenzyme, then the complex will become activated. Mission starts. And the mission is, now the complex goes and binds to the operator gene. Now this complex goes and binds to the operator gene and prevents the RNA polymerase to slide over, and thus prevents the transcription of structural genes and formation of extra tryptophan amino acid. It will prevent the transcription and further processes also. When body utilizes all the tryptophan amino acid, the extra tryptophan leaves the complex, and again protein get inactivated and the TRP operon again turned on to synthesize TRP amino acid. Thus the process repeats. TRP operon itself suppresses or represses the process, hence it is also called as repressible operon. So this is the concept of repressible operon or TRP operon. So if you have got any doubts, please ask in the comment section. Please share it with your friends. Thank you for watching, and don't forget to subscribe and press the bell icon for new notifications.